Well, good afternoon, good morning, wherever you happen to be watching this in the world. We're very excited to be announcing that we've finally got our Zonulin assay working. And so we're gonna, what I thought I'd do is introduce Dr. Brent Dorval, who's our Chief Scientific Officer, to kind of go through the testing with you in terms of a quick overview on the FIT test for some of you who aren't aware of our FIT test. And then also to give you a more detailed view on the Zonulin assay that we've developed. So in essence, really what we're going to go through is the fact that now the test has three very different elements to it, i.e. food sensitivities via the IgG1 through 4, the inflammation that we capture with the complement activation with the C3d marker, and now with a lot of excitement that we're now adding to the panel the gut permeability via two different elements. So with the Zonulin marker that we've added and developed, and Brent will talk about how that's very different from some of the others on the market, as well as Candida. So our hope is that, again, there's a lot of companies trying to make you buy more and more panels. Our approach is let's bring it all onto one test to make it much more simple for you, the provider and the patient. And then when you review the results, you've got these three distinct lines of attack in essence of the food sensitivities, the inflammation and the gut permeability, that then feeds through to how you can review the report. And then more importantly, as you think of it from a supplement sales standpoint as well. So without further ado, I'll hand you over to Brent and he can go through the FIT test and a quick overview on that, and then more importantly, the Zonulin. So thank you all for watching, and I'll pass you over to Brent, and we'll have a bit of a Q&A session at the end as well.
Yes. Good morning, afternoon, everyone. Thank you for listening in on this brief podcast. So the first thing we'd just like to discuss is, a couple of basic concepts, the innate versus the acquired immunity, and why that's important and how we measure that on the FIT test. So again, just as an overview, the immune system has three basic levels of defense, mostly against disease-causing organisms, but also chemicals and various foreign proteins, and food proteins would be included. Number one is a barrier. It's pretty obvious. You got the skin, mucous membranes, stomach acid and enzymes which degrade things, and also gut bacteria which coat the intestine and prevent penetration of molecules and also infection by other organisms. The other concept is number two, innate immunity. So we have a variety of cells, whether they're WBC, that's white blood cells. So that's the buffy coat on a serum when you spin down whole blood. So there's neutrophils, macrophages, and what they do is they phagocytize things. So they eat things and then they degrade them and prevent infection and prevent foreign molecules from gaining entry into the circulation, causing infection, etc. What's important here is complement. So complement is part of the innate immune system, and there's a molecule called C3b. It binds to bacteria, and it also forms on immune complexes. So you get C3b deposited, followed by C3d, which is the component that's measured in the FIT test. And then lastly, there's acquired immunity. Again, white blood cells, there's T cells and B cells work together. They destroy cancer cells and other infected cells, but important concept is B cells produce the antibodies. The antibodies against food antigens bind to those food antigens and then activate complement and generate inflammation. This is just a little pictogram of the FIT test technology showing you the difference between a conventional IgG only test versus the IgG plus the immune complex C3d component. So on one side, you can see the conventional conjugate generates one signal. So the antibody from the serum binds to the food antigen on the plate, and then we use a detection reagent, in this case IgG only, so that's the acquired piece, to bind to the antibody and then generate a signal. So remember, so that's all we measure here. This little green component is C3d, the inflammatory component, and in conventional tests, that is not measured. So that's very important. Now, by contrast, you look at the enhanced system that KBMO uses, and what you can see, that very same antibody binds to the food antigen on the plate. We also use an anti-human IgG 1 through 4 to generate a signal. But what we also use is an anti-C3d component that's bound into an immune complex. And what you can see is we take advantage of the signal generated from that complement or inflammatory fragment. So this test generates 2 signals, while other tests generate only 1 signal. So what you see is an enhanced sensitivity and much, much better specificity. So now let's just switch into zonulin. So where this becomes important is that foods enter the body, they make their way to the gut. And if the gut is permeable, then these food antigens can get across the gut wall and then generate immune complexes and cause inflammation. And one of the molecules that opens up the tight junctions between cells is zonulin. So what happens is an inflammatory molecule enters the gut, it binds to a receptor, which then causes zonulin, the protein, to be produced. What happens is then it's released and binds to either the same or an adjacent cell by another receptor, and it opens the tight junction. And this is occludin, those 3 little markers in between these 2 epithelial cells in the gut. So those open up and they enable the passage of these food antigens, in this case, gliadin and bacterial components, to make their way through the gut epithelium, through what's the basement membrane, and then start to generate immune complexes and cause inflammation. In the next slide, this is a little more detailed, kind of a drill down of what's actually going on, and the generation of zonulin antibody, which is very important. It's a stable marker of inflammation and the basis of the KBMO inflammatory marker on the FIT test. So what happens again, as I mentioned, in this case, gluten and gliadin, they bind to a receptor. It causes the product, goes to the nucleus, causes the production of zonulin, binds to what is called the EGF receptor on cells, the epidermal growth factor receptor. And then what happens is it stimulates what's called the actin component, just like a muscle, and it pulls apart occludin. So occludin interdigitates and closes the cells, the intercellular compartment. When that gets pulled open, now what you have is a pore that enables these antigens to get through the gut wall, and that's when things start to happen. So T and B cells work together and ultimately generate antibody. If there's food antigen in the vicinity, you get an immune complex. These things deposit very often, and then you activate complement and generate inflammation. So that's the C3d that you saw in the previous slide that is measured on this test. So we measure the antibody that's produced by the B cells, and then the C3d, which indicates inflammation, that's also generated. So you get 1 signal for antibody and 1 signal for C3d. So what this slide really ends up in is, once zonulin gets through the basement membrane and through the lamina propria, is you generate an antibody also against zonulin. Now, as we'll discuss a little bit later, zonulin protein level goes up and down, fluctuates very rapidly, very hard to get a stable measurement, whereas antibodies produced, and it can be stable for as much as 7 to 14 days. So it's very easy to get a good, reliable measurement of zonulin antibody that you simply can't do with zonulin protein. So zonulin has been implicated in many, many diseases and disease processes. So as an overview, it's a protein that's synthesized in intestinal and liver cells. It's a key biomarker for intestinal permeability. That's because if you find large amounts of zonulin, it's an indication that most likely you have leaky gut that accompanies that. It's reversible, meaning these tight junctions formed by occludin will normally open and close during the normal course in normal people. But it's when you have extensive degradation of these occludin barriers that now you have leaky gut, and then subsequently you generate food sensitivities and other diseases that are associated with these tight junctions opening. And elevated levels of zonulin are associated with, again, autoimmune diseases, celiac, obesity, rheumatoid arthritis, type 1 diabetes, etc. And in the literature, this is an active area of study, and almost every day people are finding new things about zonulin, and this list of diseases is increasing, again, almost daily. Now, as I mentioned, the problems with zonulin protein assays. Now, the big one is cross-reactivity. Cross-reacts with haptoglobin, properdin, various complement fragments, and this results in zonulin binding and results in low positivity rates. So in many of these assays, there's very, very few percent positive. Sample timing is, as I mentioned, huge. I mean, zonulin can fluctuate in minutes. So depending on when you take that sample, it could be high or low, and you really don't know where you are on that continuum. Sample type, again, can be serum, and some companies even use a fecal sample. So as you see in the next slide, we've developed a zonulin antibody assay that measures the antibody and not the protein. So this has important implications. Number one, it eliminates cross-reactivity. We use a unique zonulin protein that, incidentally, that was cloned and produced by KBMO Diagnostics, and no other company in the world has this particular protein. And remember, because of that, you don't get cross-reactivity with haptoglobin, properdin, complement fragments. So you eliminate cross-reactivity. Very important. Positivity rate, because you don't get this blocking, we get a positivity rate depending on the group of individuals. We're seeing 15, 20% positivity rate, which correlates very well with Dr. Fasano's work that shows in individuals, in groups of individuals that have certain food sensitivities or food allergies or other types of diseases, you get roughly 15, 20% or a little more positivity rate, and we get good correlation. Sample timing is not as important. So serum IgG will go up once the B cells are stimulated, and for 7, maybe even 14 days, you get a nice stable level of IgG, making it much, much easier and more accurate to measure the antibody as opposed to the protein. The sample type, we use serum or blood spot, so a dried blood spot, and we do not use a fecal sample. So we're finding that a lot of people prefer dried blood spots because it's stable, it's easy to get a sample, and when in the lab, they're easy to extract with good correlation to serum. So just a better sample all around for many people. Now, so what we say is the zonulin IgG antibody screening assay is a next generation. And what it does is it really features a unique recombinant protein, and that's very, very important. So zonulin itself, it measures zonulin IgG antibody. It's more stable and specific than the zonulin protein measurement, and it has a similar positivity rate as Dr. Fasano, especially what he's reported in the Lancet. The test format is a straightforward ELISA using serum or a blood spot. This is the sample type also used in Dr. Fasano's research work as well. So we have good correlation of results as a consequence. And we have a good team of people obviously working on this. Dr. Fasano is a very, very astute individual and discovered zonulin And I'm working with him and with other people at KBMO to move this technology forward. So I feel fortunate to be in that group. And what's important as a sales point is that the FIT 132 and 176 will include the zonulin screening assay as soon as we launch it. So that should be first, second week in March. And James can discuss that in more detail, the exact pricing on that and distribution. So just in general conclusions, we use multi-pathway detection, both the innate and the acquired immune system. So IgG is the acquired, complement is the innate. Other tests only measure acquired. Complement generates inflammation for sure. That's well known, and we measure a marker for that, C3D, of the innate immune system. Anti-zonulin antibody is a marker of gut permeability, and it's a stable marker that can be measured easily. And we use a unique recombinant zonulin, which eliminates cross-reactivity that's associated with all these other tests, which is very, very important. And with that, I'll conclude today's podcast, and I appreciate everybody listening in. Thank you.
So before we let Brent go, we'll just see if there's any questions. I've had a couple here. So one of them is, Brent, is the zonulin going to be on each of the 3 panels? So the FIT 22, the 132, and the 176?
The zonulin will be offered on the 132 and the FIT 176. It will not be offered on the FIT 22, which is just a small screening panel. So again, James will discuss the pricing and distribution of these new kits. But what's exciting is both of the larger panels will contain a measurement of zonulin antibody.
I mean, I think a good time. Someone actually asked about pricing. So you're right, Brent. What we're going to do, and it'll be dependent on your distributor in the countries that you're working in. But we're going to see a small price increase on the 132 and the 176. The good news is it'll be significantly less than if you were, as you could do today, order the FIT 132 or the 176 plus zonulin separately. So you'll see a decrease from that price, but there'll be a small increase on just the base price because we're going to include that test on there. Another one that's come in. How does this compare with other zonulin assays out there, Brent?
I think, as I mentioned, this measures the antibody and not the protein. So the protein assays suffer from problems with sample timing, and the big one is cross-reactivity. So with cross-reactivity, really, are you measuring zonulin? Are you measuring a complement fragment or some other protein like a haptoglobin? You really don't know. So cross-reactivity is really the death knell in many, many of these assays, just because you don't know what you're measuring. And the other one, again, just to hammer it home, is timing of the sample. If zonulin protein can rise and fall literally within minutes in the serum sample, it's very hard to get an accurate sample, even if you were measuring zonulin. So that compared to antibody, which is stable for a week or two. So once it rises, you can get a sample and you can measure it. So the accuracy and the specificity, so you don't have cross-reactivity, is much, much better.
Another one, let's look at some groupings around this one. Why is the FIT test better than IgG alone?
Okay, IgG alone measures only the acquired immune system, so IgG, and generates one signal, whereas the FIT test measures the acquired, which is IgG, and the innate immune system, which is C3D. So that has important implications in sensitivity and specificity. So remember, the FIT test, you generate 2 signals. The IgG only test, you generate 1 signal. So the KBMO FIT test, you get better sensitivity and better specificity.
And Brent, another one just came up as well. I mean, how many foods do you generally see are sensitive on the FIT test versus maybe some of the IgG only? Are you seeing as many, or is there some differences there?
Yeah, there are definitely differences. I mean, many of these IgG only tests, what you see is 20, even 30 positives in a large panel of foods. Whereas on the KBMO FIT test, you see probably maybe 6, maybe to 12-ish, maybe 15 at the very most. So that is because the specificity problems with IgG only tests, whereas KBMO is sensitive and specific. Now, the ramification is when you design an elimination diet and you can deal with 6 to 12 things, a patient is compliant. You generally can identify the sensitive foods. And what happens is you can manage the diet. Whereas if you've got 20, 30 things, patients are not compliant. You can't really figure things out. And many of these foods are simply non-specific to begin with. So as a practitioner, the FIT test is much more manageable than an IgG only test in most cases.
Here's one other one that's just come in. How do you compare the serum test with the blood spot? Is it reliable?
Yeah, that's one that it comes in frequently. Thank you for that one. And there's an important paper that just came out, and we'll be happy to share that with everyone. It shows that there's very good correspondence between serum and dried blood spot samples. The main thing is stability and how you extract them. That's something we do very well at KBMO. And when we look at this sort of comparison in the lab, we get well into the high 90s when you look at serum for sensitivity and specificity compared to dried blood spots. Again, that and our data is confirmed by this large study that was completed just a few months ago. So we're very, very happy to have confirmation that, again, we can offer the dried blood spot with complete confidence to our customers.
Excellent. Well, thanks, Brent, for going through those questions. And thank you all for listening. Again, we just want to kind of pass this out to everyone. And as I say, we're very excited to get the new Zonulin test out there, and it's available, as Brent mentioned, kind of in early March. And so again, please contact your distributors and they'll be more than happy to walk you through these advantages and hopefully share the slide deck with you as well. So thank you very much indeed. And thanks again for your time.